you very much for the introduction. So, as he said, the title of my presentation is The Genetic Characterization of the Multi-Drug Resistant Mannheimia Isolated from High-Risk Stocker Cattle. And as I'm sure you're all aware, Mannheimia is a significant cause and factor in bovine respiratory disease in cattle. So the goal of our group was to better characterize the genetic dynamics and resistance patterns of Mannheimia haemolytica isolated from high-risk stocker calves early in the feeding period. Now, in 2017, we published this study that kind of inspired the research that I'm going to be presenting today. So in that study, we had 169 auction market-derived bull and steer calves weighing about 229 kilograms on average. And they were processed at arrival to a stocker facility following standard industry practices, meaning they were castrated, vaccinated, dewormed, and tested for BVD. It's also important for me to remind you all that they received a metaphylactic dose of the antimicrobial tulathromycin at that time. That's a fairly standard practice in the stocker cattle industry in the United States, but it's important to keep it in mind as I present the results of that study. Then we performed a deep nasopharyngeal swab on those animals that were selected, and we submitted those samples for culture and susceptibility testing using Kirby-Bauer disk diffusion. Now, we performed, at the time of revaccination, a second sample, which occurred 10 to 14 days later, or if the calf happened to become ill before that time and was treated for BRD, we would do our nasopharyngeal swab at that time. Now, the reason that we did that is we didn't want to risk that calf becoming a mortality and falling out of the study. We wanted to make sure to include that. So if he happened to get sick before revaccination, we would go ahead and swab him at that time. Now, to show the results of this study, at arrival, we had 27 of 169 calves that were, in fact, culture positive for Mannheimia. When we look at our second sampling time point, however, we see a dramatic increase to 123 of 169 calves being culture positive. That's 72.8%. So we have an increase from 16% to 72.8% of the calves being Mannheimia culture positive. When we look at our Kirby-Bauer susceptibility by isolate, I want to keep in mind that in this particular table, we only used information if we had a CLSI-defined breakpoint for Mannheimia for these specific antimicrobials. So we did test susceptibility for antimicrobials, but I don't present that information here because we don't have a breakpoint defined. But when we look at Septifer, we see that we're maintaining nearly 100% of our susceptibility at arrival and at our second sampling time point at revaccination. However, when we look at the fluoroquinolone antimicrobial enrofloxacin, we see that 96% of those isolates were susceptible at arrival, and nearly 100%, 99.2 in fact, were resistant at our second sampling time point. When we look at fluorphenicol, we don't see quite as dramatic a picture. We do see that 100% of our isolates were susceptible at arrival, and about a third of those remained susceptible at our second sampling time point. But at revaccination, we had 68.3% of them being resistant. When we look at the macrolides together as a group, we see fairly high levels of susceptibility at arrival, and then we see anywhere from 98 to 99% resistance at our second sampling time point. So that was really interesting. When we look at co-resistance by isolate in this study, we see that we only had 3 isolates out of 79 that were resistant to 2 or more different classes of antimicrobials, and those are indicated here with underlines. So we have enrofloxacin, and then we have our macrolides together as our group. When we look at the second sampling time point, we had 363 isolates out of 366 that were resistant to 2 or more different classes of antimicrobials. I'm not going to read all of those off to you, but you can see that we have quite a few different patterns. In other words, 3.8% of our arrival isolates were co-resistant, and 99.2% of our revaccination isolates were co-resistant. So that led us to ask some new questions, and we came up with a list of new objectives moving forward as we continued with our work. First, we wanted to know what the genetic relationship was between those Mannheimia isolates at arrival and our second sampling. We wanted to know which resistance genes were present at arrival and second sampling, and we wanted to assess the agreement between our antimicrobial susceptibility profile and our genotype. And finally, we wanted to know if there was resistance gene-bearing mobile genetic elements present in those isolates that were multi-drug resistant. So I went ahead and extracted DNA from the isolates from calves that were positive for Mannheimia at both time points. That was a total of 20 calves, yielding 48 isolates. 26 of those isolates were from arrival and 22 were from revaccination. Now you think to yourself, okay, how does that make sense? Well, if a calf was culture positive at one time point and we could get multiple colonies from that calf, we wanted to make sure to include a wide variety of them. So we had a maximum of 3 isolates that would be selected from that plate, only if they had a unique susceptibility profile. Then I did whole genome sequencing with Illumina NextSeq, and we had an outside consultant do our initial data analysis for us. So to assess the genetic relationships between our isolates at those 2 time points, I uploaded the sequences to CSI Phylogeny, which has a web interface to generate an alignment. Using that alignment, I made a phylogenetic tree using RAXML, and I used Mannheimia M42548 as our reference sequence. This is the phylogenetic tree that was produced. Each of the different colors indicates a different clade. So we have the top 3 clades indicated in purple, green, and blue being entirely arrival isolates. Any that you see in black are commonly used isolates in research that I just wanted to include so that we could see where my isolates fell in relation to commonly used isolates. If we look at the red clade here, though, we do see that it is predominantly revaccination isolates, and that's indicated by R. When we look at this top one, however, we do have one arrival isolate falling into this group genetically. Now, the bootstrap support for each of these clades was 100%, and the bootstrap support for most of the major branches was greater than 90%. When we do get to the bottom few isolates there of the red group, we have poor bootstrap support, and that's because those isolates were all in fact clonal. So we don't have good bootstrap support for that. For those of you guys who make phylogenetic trees on a regular basis, you'll understand that. So when we look at the resistance genes that are present in our isolates, these are the ones that we found. I won't read them all off to you, but we do have resistance to phenocols, tetracyclines, sulfonamides, aminoglycosides, beta-lactams, macrolides, and fluoroquinolones represented here. The way that I obtained all of this information was I blasted it against the CARD database in MegaRes, and I did in fact hand curate those to eliminate any false positives and to assess them for gene links. Some of these will come up as hits, but will only be partial sequences, so any that were just like a gene fragment were eliminated. We do have 3 resistance genes present in our arrival isolates, TET-H and TET-R. Those were present in the same isolate. And then we had another isolate that actually had BLA-ROB1 as well. And at revaccination, we had a total of 14 different resistance genes and 3 fluoroquinolone point mutations present. Looking at those specific point mutations a little more closely, we see a few different things, but it's important to remind you all that you do have to have a point mutation in both GYR-A and PAR-C to see fluoroquinolone resistance. And all of our resistant isolates had a change from S83F, from S83 to F, and at location 87, we had a D to N change in GYR-A, and for PAR-C, we had an E84 to K change, as indicated here. So to look at the agreement between our antimicrobial susceptibility profile and our genotype, I used the antimicrobial susceptibility data from the previous study to assess the agreement. And just to remind you all again, that was based on Kirby-Bauer diffusion. And we only used antimicrobials with CLSI to find breakpoints. We had a total of 7.7% of our arrival isolates that were resistant or intermediate, and all of our revaccination isolates were multi-drug resistant. Just to explain this table a bit more clearly, we have the genes listed here on your right. We have the FLO-R, TET-H, and TET-R for ERM42, MSRE, and MPHE for each of the macrolides. Now, to be considered concordant, they have to be both susceptible and negative, and resistant or intermediate and positive. And the opposite is true for discordant. We do have one isolate that was resistant or intermediate and negative for flow R. And we have 7 isolates that were resistant or intermediate to telmicin and negative for any of the macrolide resistance genes. When we look at telathromycin, we see a similar story, but it's only 5 isolates that were resistant or intermediate and negative for a given macrolide resistance gene. So in terms of our fourth objective, are there ICEs present in our isolates? And the answer is yes. So here I have illustrated one of those integrative conjugative elements. This is from isolate R1, 20-1, meaning this calf was a revaccination calf from the first week of arrival, year tag number 20, isolate number 1. The particular ice that we found was very similar to ICEP MU1, which is from Pastorella multacida, and it does have some similarities to other ices as well. We have 2 resistance gene regions indicated here in the red boxes. Those contain aminoglycoside resistance genes, FLOR, macrolide resistance genes, and tetracycline resistance genes, as well as the beta-lactamase gene OXA2. And all of our revaccination isolates, the sequences are in fact positive for ICE-associated genes, INT2, ICE relaxase 1, and PARB being some of those genes. And in fact, that arrival isolate that fell within the same clade as the revaccination isolates, if you recall the phylogenetic tree, did in fact have ICE relaxase 1. It has integrase 1, PARB, TET-H, and TET-R. So it's very likely that that isolate also has an integrative conjugative element with fewer resistance genes present in it. In conclusion, our isolates fall into 4 clades, with our arrival isolates in 4 clades and our revaccination isolates in a single clade. We had a number of resistance genes present, as well as a number of fluoroquinolone point mutations present in our resistant isolates. And the concordance for FLOR and fluorphenicol was not perfect. This is also true for telmicin and telathromycin. And we do, in fact, have integrative conjugative elements present in our multi-drug resistant isolates. These are my acknowledgments, everyone in my group, as well as the Georgia Beef Commission and the Dr. Charles Dobbins Fund, and Mr. Keith Mitchum being the stocker operator who allowed us to sample his calves. Thank you.